Welcome to the C2 Pipeline Innovation and Curiosity Center Sheep Brain Dissection Lab. After completing this lab, you should be able to do the following: understand the locations of various parts of the brain, understand the planes of orientation, understand vocabulary relating to the brain, safely dissect a sheep's brain. Safety in the lab is very important. You should wear gloves, safety glasses, and a lab coat if available at all times during this lab. You should also have your sleeves rolled up and your hair pulled back. Be extremely careful with the dissecting tools as they are very sharp. Never walk around the lab with tools in your hand. Let's take a look at the various locations of brain structures. Anterior, or rostral, means the front of the brain. This is the part of the brain that does not have the brainstem. Posterior, or caudal, means the back of the brain. This is the part of the brain that does have the brainstem. Medial is at or towards the middle. Dorsal means on top. Ventral means the bottom. Lateral means from side to side. The brain can be divided into many different planes. There is the frontal or coronal plane, which divides it from front to back. It can divide the brain in any location as long as it divides the brain from front to back. So it could be toward the front of the brain, or you could be making the cut toward the brainstem. It doesn't matter the location again as long as it divides it from front to back. The sagittal planes divide the brain into the left side and the right side. The most important sagittal plane is illustrated is the mid-sagittal plane. Any plane that is parallel to the mid-sagittal plane is also a sagittal plane. The last planes are the horizontal planes. They divide the brain from top to bottom. These planes are illustrated in the photos. The first portion of this dissection lab will be a detailed examination of the brain's surface. No actual cutting of the brain is required for this portion of the dissection. You will be instructed when you should be making any cuts. At this time, we're just examining the brain. As you proceed to identify the listed parts of the brain, note their structure and how they are related to other parts of the brain. At this time, we will pause to get our materials and our brains for this lab. First, examine the exterior of the brain, or the outside of the brain. The brain, as well as the spinal cord, is surrounded by 3 layers of membranes, which consist of dura mater, the pia mater, and the arachnoid layer. You may be able to see dura mater, or the tough outer layer, on your brain. This layer probably mostly has been removed, but some of the dura mater may remain near the base of the brain. The pia mater is the delicate innermost membrane enveloping the brain and spinal cord. The arachnoid layer is much thicker than the dura mater and the pia mater and is attached to the inside of the dura mater and surrounds the brain and spinal cord. Next, we are going to locate the area referred to as the brainstem. This area is made up of the pons, the medulla, and the cerebellum. We're also going to find the root where the pituitary gland was attached to the brain. After the facilitator pauses this video, you should turn your brain so that you are looking at the ventral side or the bottom side of the brain in order to find these items. We will pause at this time. While examining the ventral surface of the sheep brain, you should find a pair of olfactory bulbs that may be seen, one under each lobe of the frontal cortex. See if you are also able to identify the pons, pituitary stalk, mammillary body, and optic chiasma. Every brain will look different, so don't fret if you can't identify the following structures. We will pause to attempt to locate the labeled parts of the brain. When the facilitator pauses this video, you will turn your brain so that you are looking at the dorsal or top part of the brain. You will then locate the spinal cord. Are you able to see the cerebral hemispheres and the longitudinal cerebral fissure? Let's pause to look for these items. When we pause the video, we'll look for the optic nerves as well as the pituitary gland. Let's pause at this time. There are 4 lobes of the brain: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. When we pause this video, we'll take a moment to see if you can identify the 4 lobes with your partner. Let's pause now. Woohoo! Let's move on to dissection. For the following part of this movie, please wait for directions from the instructor before making any cuts. The instructor will pause the video and give any necessary further instructions before any cuts should be made. When the video is paused, you will make a mid-sagittal cut where the red line is. Make sure your brain is level on your dissection tray, and then you will cut along the longitudinal fissure. Once you make your mid-sagittal cut, your brain should look similar to this photo. We will pause to make the cut at this time. If you look closely, you should be able to find the cingulate gyrus, the lateral ventricle, and the third ventricle. We will pause to see if you can identify these structures with your partner at this time. Now, you will carefully make cuts 12 and 13 as shown in this picture. Please note the cuts are on a diagonal and they cut through specific areas of the brain. Take a very close look at this picture so that you are sure where you should be making these cuts. We will pause at this time to make the cuts. Hopefully you were successful in making your cut. If you were, you should be able to find the following. The basal ganglia is made up of 3 different structures: the putamen, the globus pallidus, and the caudate nucleus. Let's pause at this time to locate these structures. Still examining the piece that you cut out of the brain, with your partner, find the hippocampus, the lateral geniculate, the medial geniculate nucleus. We will pause at this time to find these structures. Our dissection of the brain is complete. Let's pause this video to discuss the questions on the screen. This concludes the sheep brain dissection lab. We hope you have enjoyed yourself.